Hi, everyone. Thanks for tuning in to my talk. My name is Jianfei Shao. I am a PhD student in Maharali Lab in Department of Integrative Biology in University of Guelph in Ontario. My study interest lies with evolution of plant root systems, and today I'm going to talk to you about some of the findings from the first chapter of my PhD. As we know, root is an integral part of all vascular plants, critical for anchoring the plant, storage, as well as the transportation of soil nutrients and water, which ensures plant growth, survival, and reproduction. In nature, there seems to be a lot of variation in root traits among different plant species, families, as well as among different plant growth forms. As you can see on the illustration, different plants can vary greatly in terms of their below-ground root characters. They can differ in root biomass allocation, root depth, or root branching, and many other morphological traits. The diversity in plant root morphology leads to differences in plant root functions, and one of the major functions of root is the acquisition of nutrients. One common categorization of root morphology are the trade-off between maximizing plant growth and nutrient acquisition versus survival and resource conservation. The root systems that have higher absorptive capacity are roots with more surface area. They tend to be finer, cheaply constructed, and more branched out, and they have high nutrient acquisition capability. They're depicted on the pictures on the left. While roots with lower absorptive capacity are roots with less surface area, they tend to be thicker, less branched, and generally with longer lifespan, and these roots have less absorptive capacity, and they're displayed on the pictures on the right. Phosphorus and nitrogen are both essential nutrients, which are vital for plant growth, and their deficiency is common across the globe and widespread among natural ecosystems. They're the main factors limiting plant growth. Studies have shown previously that variation in nutrient availability can lead to variation in plant root expression. My study is focused on phosphorus availability because, as you can see in the map, phosphorus limitation, which is in blue, is more widespread in temperate and tropical areas where most people live, and it also impacts a larger diversity of plants and ecosystems. In addition, phosphorus is a more immobile nutrient that is more inaccessible to plants. This could mean that phosphorus availability applies more selective pressure on root functional traits. Adaptations such as increased plant allocation, increased root depth are found to be associated with phosphorus deficiency. By extension, if limitation in phosphorus results in more root allocation, it could also naturally lead to the expression of roots that have higher absorptive capacity that are better at acquiring soil phosphorus. So phosphorus availability can be a major factor driving evolution of plant root systems. Current studies done on how phosphorus affect root traits are all done on crop cultivars, but not on natural populations displaying natural diversity. In addition, no study have linked root traits variation with plant performance and measure selection under high and low phosphorus settings. And that brings me to my study. So the root trait that my study chose to focus on are morphological traits that are not often studied in previous research on how phosphorus affect root traits. So when we talk about traits with high absorptive capacity, we would be referring to roots with high specific root length, SRL. So this also means high root length per unit biomass, higher degree of root branching, and lower root tissue density, RTD, which is measured by biomass per unit volume. So this also means that the root is more cheaply constructed. So these sets of traits may evolve under low phosphorus to achieve higher nutrient acquisition. On the other hand, when we talk about low absorptive capacity roots, it would be the opposite. So we will be talking about low specific root length, lower root branching, and higher root tissue density, or RTD. These traits could evolve under high phosphorus conditions. The main question that I'm exploring is, are root functional traits being selected for under low and high phosphorus conditions? If a certain trait is adaptive to an environment, then the plants expressing these traits would have better performance in said environment. In my study, selection on a trait under a specific phosphorus environment is given by performance differential, which is calculated by the slope of regression between standardized root trait, which is an independent variable, and relativized plant biomass, which is the dependent variable. For these hypothetical prediction regressions shown here, I'm only using SRL as an example. So under high phosphorus, plants with roots with less absorptive capacity will be favored. We should see lower SRL yielding higher plant performance, and thus see selection for lower SRL under high phosphorus conditions. Under low phosphorus conditions, plants that produce roots with higher absorptive capacity will perform better, and we should see selection for higher SRL under low phosphorus environments. The subject species of my research is the Mediterranean legume Medicago truncatula. The plant has short generation time and has many well-defined naturally inbreeding populations that vary significantly in root trait expression, verified by a study previously done in our lab. So for my study, I have chosen 34 distinct natural populations from the Mediterranean area. Plants from all 34 populations were grown in University of Guelph, phytochron greenhouse for 8 weeks. They were treated with low and high phosphorus fertilization treatments. Plants were harvested after 8 weeks with shoot and root biomass measured, and root functional traits were later analyzed. So here are my findings. These are reaction norm plots where we have fertilization treatment on the x-axis with low and high phosphorus, and y-axis represent the trait of interest. So these lines represent how more than 30 populations responded to low and high phosphorus applications in terms of plant total biomass and root to shoot ratio. High phosphorus increased plant growth and reduced root to shoot ratio compared to plants under low phosphorus. This is in line with our knowledge that phosphorus tend to stimulate plant growth, and plants under phosphorus limitation tend to invest more into roots. As you can see, for both plant growth and root to shoot ratio, the population term is always significant. So this means plant biomass and root allocation differ significantly among populations. This is true in both low and high phosphorus environments. The difference among population, as you can see, can be up to several folds. The significant P by E term, or population by environment interactive term, suggests that populations do differ in their response to phosphorus in terms of plant biomass and root to shoot ratio, which is indicated by the crossing of some of these lines on the graphs. For root functional traits, I found that higher phosphorus led to a reduction in SRL, which is in line with the idea that under high phosphorus conditions, plants should express root traits with reduced absorptive capacity, which is lower SRL. I also found that higher phosphorus led to an expression of the higher root branching. This seems contrary to the idea that under high phosphorus, plants should be expressing reduced absorptive capacity traits due to the high availability of phosphorus, and that should be lower branching. As for root tissue density, RTD, phosphorus did not have an effect, suggesting that RTD is not as responsive as traits such as SRL and branching to contrasting levels of phosphorus. Of course, we're still observing large genetic differences in all of these traits among different populations, as the population terms are always significant with very low p-values. Let's take a look at selection results. In this graph, I have selection differential on the y-axis. A negative selection differential means selection for reduction of the traits, and the star denotes significant selection. All three traits are under significant selection in certain phosphorus environment. There is selection for reduced SRL under high phosphorus, which is in line with my prediction that plants with lower absorptive capacity should be linked with better performance under high phosphorus. Also, this is consistent with the root plastic response data shown previously. There is selection for lower branching under high phosphorus. This agrees with my selection prediction but contradicts plasticity result, which showed higher branching under high phosphorus. The detection of selection for reduced branching here under high phosphorus is meaningful because this establishes a linkage between lower branching and better performance under high phosphorus. For root tissue density, we have detected selection for lower RTD under low phosphorus, which is in line with the idea that higher absorptive capacity is favored under low phosphorus conditions. So what are the implications of these findings? Well, our results show that there is significant selection for SRL, RTD, and root branching in certain phosphorus environments, which means that the expression of these traits are linked with plant performance. This suggests that phosphorus availability is likely a main factor driving the evolution of plant root functional traits. The direction of selection is largely in line with my predictions, which gives us useful tools to predict the longer-term evolution of plant root systems. For instance, an increase in agricultural fertilization activity is known to cause a higher level of phosphorus deposition in surrounding ecosystems. So that could lead to evolution of root traits that exhibit lower absorptive capacity. Finally, selection is found to act on all of these root traits and not just on any one of the traits. So this is indicating that selection under contrasting phosphorus environments is multidimensional, and all of these traits could be key root traits in determining plant capacity for phosphorus acquisition. And with that, I will wrap up my presentation. I would like to thank my founding sources and members of the Maharali Caruso Lab, and all staff who work at University of Guelph Phytoton for all of their help, and members of my advisory committee for their advices. I am happy to take any questions, and also feel free to email me for any questions and further discussions on the email displayed here. And thank you very much for listening.